From Diamond Light Source, this is the Diamond Podcast.
Welcome to the Diamond Light Source Podcast with me, Mira Senthilingam. This month, we're probing down to the atomic scale as we get an insight into the world of structural biology. We'll be finding out just what structural biology is, the molecules it can enable us to see, and investigate how visualizing molecules such as proteins can help find treatments for conditions like preeclampsia.
We've shown specifically the change of angiotensinogen to the more active oxidized form takes place in preeclampsia. It opens prospects for the treatment of what has otherwise been a very resistant condition. That is the high blood pressure of pregnancy that is the accompanying and prime feature of preeclampsia.
Cambridge University's Robin Carroll will be discussing the role of the protein angiotensinogen in hypertension and preeclampsia, and how knowing the structure of this protein could result in a whole new class of drugs against the conditions. We'll also be hearing about the proteins involved in repairing our DNA and guiding the development of our nervous system. So all that insight into the proteins that keep our bodies functioning, plus the latest news and events from Diamond, coming up in this November edition of the Diamond Light Source Podcast.
The Diamond Podcast. For more information, look us up online at diamond.ac.uk/podcast.
Investigating and visualizing the structure of molecules such as proteins and DNA can provide a great insight into just how our cells, and therefore our bodies, actually work. And knowing just how we work can help us solve the problems that arise when something stops working, a knowledge that the field of structural biology is helping to provide, as Gwyndaf Evans explains.
So structural biology is really a subset of biochemistry and molecular biology and biophysics that really aims to get a structural understanding, hopefully at the atomic level, of the most sort of active molecules in cells and in the body in general. And they're principally proteins and nucleic acid, things like DNA, that are responsible for the majority of the functions going on in the body and indeed in any organism. The importance of understanding the structure is then to try and relate that to how these molecules function. Then we can understand how mechanisms of diseases come about, and we can think about designing drugs and curing disease through an understanding of that.
So the key really here is to visualise and see the structure of these particular proteins and molecules. So we're now at the Microfocus Macromolecular Crystallography beamline, where you are the principal beamline scientist. So tell me a bit more about this beamline. It's an interesting setup here. How does this process actually work to visualise a protein structure?
Generally, the method we deal with here is fundamentally crystallography. It's X-ray diffraction from crystals. So it relies on scientists in the home labs producing crystals of the molecules that we're interested in sort of looking at. What Diamond Light Source does is produce these incredibly fine beams of X-rays that we can then use to sort of hit these crystals. What happens when an X-ray beam interacts with any sort of ordered lattice or a crystal is that it sort of generates a sort of a very intricate pattern of spots that we have to sort of record. We measure those on sort of large area detectors that we put behind the crystal, and sort of analysing those spots gives us the ability to sort of reconstruct what those molecules look like at an atomic level, hopefully.
Okay, well tell me a bit more about the setup here. We're about 50 metres away from the actual synchrotron now, in the sample area. So there's a series of metal boxes, but we're out by the final one, where I imagine the beam actually comes out to hit the sample. And now the sample's actually placed on a sample pin, which, looking at it, it's minuscule. It's less than a millimetre wide.
Yeah, if we looked at this under a microscope, the end of it would be a tiny little nylon loop, a fifth or a tenth of a millimetre in size. And they have to sort of use these loops to literally fish the crystals out of a small drop that has been used to sort of grow the crystals in. They'll then immediately freeze those crystals in liquid nitrogen to very low temperatures, which is necessary really to make them survive in the X-ray beam a little bit longer than they normally would. Then they'll come with anything from maybe 10 or 20 up to several hundred crystals to the synchrotron. One by one, they'll be mounting those crystals using a robot The sample is actually mounted on this sort of rotation, this motor that just rotates the crystal around as well. So the actual experiment that's going on here is something called the rotation method, where while the X-rays are hitting the crystal, we rotate it around, and that allows us to measure really a complete sphere of data, as we call it. So just
giving you a full 3D structure.
Absolutely, that's exactly right, yeah.
So you've got this sample set up here, the beam is hitting the sample, and just about a foot away, you've got a large, maybe half a metre squared kind of box, which I'm assuming will pick up the scattered X-rays then?
Yeah, this is a sort of solid-state pixel array detector, and it's built up of many silicon sensor modules, loads of those put together to make this sort of square-looking face, if you like. This thing works at very high speed, so what we can essentially do is rotate that crystal and just keep it rotating, and this thing virtually records a movie of these X-ray spots that are being generated or being produced by the crystal and the X-rays. That data is then just analysed offline, so computer programs just will run through that stuff and work out what the molecule looks like from that.
What are you actually looking for when you get, say, a picture of a protein, say?
What we actually sort of see is sort of a cloud representation of the electrons. We're not really sort of seeing positions. We get something called an electron density map. Atoms are just sort of surrounded by clouds of electrons, and it's the X-rays that interact with those clouds of electrons that we're measuring. So the final sort of structure, the final model that people are building up is really a point representation of where atoms are within that structure. One of the biggest applications is obviously now in the drug industry in terms of pharmaceuticals and drug discovery. But recently, what synchrotrons and what these new sort of developments, these sort of big establishments and fine beams have given us the ability to do is work on larger and larger and more complex structures.
What are the challenges perhaps now, and what perhaps maybe is this beamline designed to overcome with particular challenges?
The case for building this beamline really came out of wanting to develop a better understanding of particular classes of proteins. Firstly, membrane proteins, also viruses, and more generally then these large macromolecular complexes. The importance of membrane proteins as a class is that as the potential source of targets for drugs, they represent virtually half of all of the interesting targets. Over the last sort of 50 or 60 years, we've built up a library of protein structures which now contains getting on for 60,000, possibly more protein structures, but the structures of membrane proteins are massively underrepresented in that library. We only have, say, just over 200 membrane protein structures that we've solved. The way structural biology is evolving now is by trying to build up a more comprehensive picture of how these smaller structures that we've sort of solved over the last 50 years interact with one another, how they go together to form these larger structures, and how then this complex picture of many proteins and complexes interacting with one another, how they go to form the cell, basically a living cell.
Gwyndaf Evans, Principal Beamline Scientist on Diamond's Microfocus Macromolecular Crystallography Beamline. Now, as Gwyndaf mentioned, more insight is needed into the structure and function of cell membrane proteins. And one scientist working on a class of these involved in our neural development is Yvonne Jones from the University of Oxford.
So for the last couple of years, I've become particularly interested in the cell surface receptors that are responsible for allowing cells to be guided to the right positions in the body, and having got there, stay there. So they're really determining the balance between adhesion and mobility. And there are several families of these that are particularly well studied at the moment in the nervous system because of their roles in the development of the nervous system, in particular in guiding the growing axons from nerves to their correct location during development. One of the big families that's turned out to be very important in this is made up of the semaphorins, the proteins that come in and trigger the activity of the receptors. And the receptors are called plexins. So particular flavors of semaphorins bind particular flavors of plexins. And in general, the result of that binding is that a cell moves away from the direction that the semaphorin is coming from. So it's a repulsive signal.
And where do these semaphorins actually come from then? So how are they released in the first place to then come along and bind to the plexins and repel the cells?
They're encoded in our genome, and At appropriate times, their expression is switched on, and many of the semaphorins are actually still attached to cell surfaces, but some are secreted. The ones so far that I've worked on are naturally cell-attached ones.
And now your more recent work has been actually trying to look into the structures of these proteins, so these receptors and these molecules.
I want to actually look at the atomic level details of how a semaphorin interacts with a plexin and what it is about that interaction that allows the plexin to know that it should be signaling.
How do you use DIAMOND then? How do you use the synchrotron to actually then visualise the interactions that are taking place?
So we've been using several of the macromolecular crystallography beamlines, the MX beamlines, to see the detailed shape and surface characteristics of the plexin receptor and the semaphorin, and to understand how the two things recognize each other very specifically and lock together. The exciting thing is that we now know that a semaphorin isn't just one molecule, it goes around in pairs, and that actually turns out to be really important to the way it functions. The plexin, on the other hand, is tending to be just by itself. Once the semaphorin comes in and binds it, it will actually serve to pull together two copies of plexin. And in fact, we think what's happening is that the plexins then start to actually cluster to quite large groups. And it's those large groupings of plexin that are needed to start the signal going.
So these pairs of semaphorins are what are really basically attracting, well, more than just a pair of plexins, so lots of plexins grouping together to then activate the signal.
What we can see initially, what we can see in the crystal structure, is a pair of plexins. But once we start looking at things back in the context of the cell surface, we and others suspect that actually they're then beginning to form whole clusters, whole crowds and it's that becomes important in sending the signal.
Knowing this then about this interaction and just how it's controlled, could this be perhaps used for any medical applications?
If we can understand how these systems work, then there may turn out to be instances where we want to go in and very specifically influence that signaling. One example where we think it may be important to be able to go in and stop the semaphorin plexin interaction is where we want to be able to regenerate nerve growth where there's been damage, say, to the spinal cord. This isn't the only system that it might be important to influence, and we're working on several such systems in the lab to understand how these very specific protein receptor interactions could potentially be targeted with the design of new drugs that might then allow nerves to more easily grow back to repair damage in things such as spinal cord injuries.
Yvonne Jones, Professor of Protein Crystallography at Oxford University.
Did you know that diamond is so powerful it produces more light than 100 million suns?
You're listening to the Diamond Light Source podcast, and this month we're probing down to atomic scales to visualise the structure of some of our body's most crucial molecules. And still to come, we discover the workings of a protein involved in hypertension and preeclampsia, and find out how understanding the workings of this protein could provide a whole new class of drugs against the conditions. But before that, let's join Sarah Boundy from Diamond's communications team for a roundup of the latest news and events from Diamond, starting with good news in the form of funding.
Yes, last month we found out that Diamond's going to receive capital funding for its Phase 3. It basically means we'll get £69 million from the Large Facilities Capital Fund over the next 4 years. That means we're able to build 10 additional beamlines by 2017, and that will bring the total to 32 and maximise Diamond's potential to support UK science and industry.
But Phase 2 is still currently in operation, isn't it, as well?
Yes, so work continues on the final 4 Phase 2 beamlines, and they'll be ready by 2012.
So what's Phase 3 going to offer?
Well, after consultation with the scientific community, we've identified the first few beamlines. There'll be an innovative macromolecular crystallography beamline to enable structure determination at longer wavelengths. We want to improve imaging capabilities to enable imaging of materials at levels of detail unprecedented for the UK. There will be more facilities which will allow the study of electronic structure of the most complex and intellectually challenging materials of the physical world. And with Phase 3, we also aim to increase high throughput access.
As well as looking into the future, though, Diamond has been around now since 2007, and you've reached quite an important milestone in terms of the papers that have been published as a result.
Yes, we're really excited to reach our 1,000 publications milestone. So we actually went into operation in January 2007, and in that time we've been producing peer-reviewed journal and conference papers. They're published by researchers using experimental data collected at Diamond, and also by our own staff, scientists and technicians.
So 1,000 publications in just under 4 years, that's quite impressive actually.
Yeah, it's really good. I mean, we started with 7 beamlines, and whilst producing papers, we were also adding on more beamlines, so we have 18 now, and it's with those 18 that we're able to produce these papers. And the most recent paper is the first user paper from our latest beamline to go into operation. That's B18, the Core XAS beamline. Other highlights include Josep Soler Suso's work on using infrared light to identify cancer cells. There was also a paper about moving closer towards hydrogen-fueled cars by improving gas storage. And Parkinson's research, engineering materials. It covers a lot.
Many of which we've covered in the podcast before, actually, as well. And these are all available online as well?
Yeah, we have a publications database on our website, so anybody can just have a look at that and see the papers we've produced.
And while sticking with the website as well, people can also go there to find out more news.
Yes, we've just released the Autumn 2010 edition of Diamond News. That's our newsletter that we release twice a year. This issue covers understanding nickel and how it bonds with its surrounding elements that will aid with its extraction. There are updates to existing beamlines, stories on modifications to the storage ring for upcoming beamlines. And we had a really busy summer, so the newsletter also gives a roundup of events and workshops over the last few months. And we cover a particular piece of research that led to a breakthrough in preeclampsia. Preeclampsia is a condition that leads to the death of 10 women and 1,000 babies in the UK every year. Researchers from the universities of Cambridge and Nottingham recently published a paper in Nature which has identified the processes involved in the condition.
And well, one of those researchers was Robin Carroll, who we'll actually be hearing about later on in the podcast. But just to finish off, really, Sarah, there must be another Diamond Day coming up.
Yeah, our Inside Diamond open days are as popular as ever, and the places are just getting snapped up. So the next available date is actually 26th of March in 2011. And what people can do if they come along to Inside Diamond is they hear a talk about the facility and its different applications. They then get to go on a tour and actually look around and go inside the machine and meet some of our scientists and engineers. So it's a really good day out.
Thanks, Sarah. Sarah Boundy from Diamond's communications team, who'll be back in the next edition with more news from Diamond. But in the meantime, visit diamond.ac.uk for more news and updates, and also to sign up for the March Inside Diamond Day before it books up. Now, moving back to the visual world of structural biology, we now focus on the repair of our DNA. Due to various factors such as UV, our DNA is constantly being damaged, and organisms need to repair that damage on a regular basis in order to survive. There are many proteins involved in this repair, one of which is XPD, a protein that Jim Naismith at the University of St Andrews has been looking into.
The protein that we studied is a protein called XPD, and what it does is it's a helicase, and that helps you unwind the DNA around a damaged lesion, and that's one of the very first steps in repairing DNA.
If this protein, for any reason, becomes dysfunctional, what are the consequences?
In humans, there are a number of diseases. The most famous, I think, or infamous, is the so-called Children of the Moon syndrome. These are children who have to be kept away from sunlight because they're unable to properly repair their DNA damage. What that means is these children often develop cancers and or fairly other significant illnesses. There's also other phenotypes to do with hair. You get a brittle hair disease, and you also have some cases deformities during development.
So what are the malfunctions almost that happen in these proteins that result in them not working? So what happens in these diseases?
Well, you're unable to unwind the DNA and start the repair, and what we were able to show is that the mutations for one disease cluster in one part of the protein, and the other phenotype, the brittle hair disease, clusters in a different section of the protein.
How have you set about looking at the structure then of this XPD protein?
Humans are quite similar to bugs called archaea. There are 3 trees of life, if you like. So bacteria tree of life, the eukaryotic tree of life, which is us, or we're in that tree. And then there's a third branch called archaea. And archaea and eukarya diverged later than the split from bacteria. So what that means is that we are actually quite similar in some ways to these archaeal organisms, and in one of the ways we're similar is in DNA repair. Now, the human enzyme is extraordinarily difficult to work on. It's longer, bigger. The archaeal equivalent of XPD is much more tractable to biophysical methods, and so we were able to clone that, purify it, crystallize it, and determine the structure at the diamond synchrotron. But the key residues that are mutated in human disease can be mapped very easily on to the structure from the archaeal organism, and in fact you can test those by biochemical methods to see if you can recapitulate defects in the protein. So
having looked at the structure of this protein then in the archaeal versions of the proteins, what have you found about the structure of this particular protein? We
found that one set of mutations clustered in what's called the ratchet. Helicases work by expending ATP, and as they do so, they drag a duplex DNA over its space and break it up into separate strands. And so one set of mutations obviously exert their effect by working there, and that's actually the interface between 2 parts of the protein. The other set of mutations are distant from that, and what they will do is affect the ability of the helicase to recruit other proteins into the cluster, because in all organisms, but especially in higher organisms, such a complicated thing as DNA repair often brings other proteins in, and you get these multi-protein complexes. And in fact, the other set of mutations cluster exactly where you would predict other proteins would dock. Quite different biochemical phenotypes. One will inactivate helicase activity. The other one, the helicase activity is normal, but what it does is it stops that helicase being able to recruit other proteins.
So there are 2 factors really, so it's a 2-step process almost in this first early stage of DNA repair.
Yes, and if you have the inability to unwind the DNA, you get the very severe illness, the cancer, the children of the moon. If you have the ability to unwind the DNA, but not to bring other proteins to the party, you get the other very severe but not quite as severe diseases. Brittle hair sounds like, oh well, you could live with that, but actually there's actually a lot of abnormalities that go with that are very significant.
So what real kind of insight has this structural insight really provided in terms of understanding the diseases, and what other insight could be achieved through this?
So what the structure enabled us to do was to segregate why one set of mutations caused a particular disease and another set caused an apparently totally different disease. And so that wasn't known. You couldn't segregate them before, but with the structure you can. Now, the fact that the cause of one of the pathways only affects the recruitment of other proteins and leads to, in some ways, a more subtle, but as I emphasised, a rather serious illness, that may help us disentangle, or may help others disentangle, the pathway involved that goes wrong in DNA repair in those people, and that type of pathway that goes wrong in DNA repair is often seen in cancer. So these mutations may allow or may permit some guidance to see what other mechanisms are involved in DNA repair and possibly help in understanding cancer.
Jim Naismith, Professor of Chemical Biology at the University of St Andrews. Now, that's almost it for this month, but let's have one more bit of structural insight. This time into a protein which could help in the fight against hypertension and preeclampsia. Both of these conditions are unfortunately quite common, with 10 women and 1,000 babies dying of preeclampsia in the UK every year. There are various factors involved in inducing hypertension: our heart rate, the volume of our blood, and also the constriction of our blood vessels, causing them to narrow and therefore increase our blood pressure. This latter cause is what Robin Carroll from the University of Cambridge is looking into, and he told me more about the proteins behind this constriction.
A major factor is a peptide hormone called angiotensin, which directly results in a tightening or vasoconstriction of small blood vessels. Angiotensin originates from a large protein that circulates in our blood called angiotensinogen. The hormone is released by an enzyme from the kidney called renin, and the renin cleaves off the terminal part of this circulating protein, angiotensinogen, to give this very small peptide hormone, angiotensin.
You've been looking at the actual structure of this angiotensinogen protein to see just what happens to it, I guess, to release angiotensin.
Exactly, and there was new information, and in this case, 2 surprises that gave us an insight into processes relating to the control of blood pressure.
So yes, I'm intrigued. What did you find? What were these surprises?
The first and very satisfying finding, which was that angiotensinogen is far from being a passive source of the hormone. It interacts in a very active way with the enzyme renin. The cleavage site that renin attacks to release angiotensin is buried within the angiotensinogen molecule. And we demonstrated this, and also able to demonstrate the way that the interaction with renin results in the cleavage site becoming accessible, changing from the interior of the molecule to the exterior of the molecule. So we're now seeing that angiotensinogen plays a active and positive part in what is the initiation of the main process controlling blood pressure.
What actually happens to it for it to perhaps become exposed, say? There's also a second surprise.
The surprise to us and to all was the presence of a hidden switch, a fine-tuning mechanism that controls the activity of the interaction between renin and angiotensinogen that releases the hormone. And this switch is based on a disulfide bond, a bond between 2 sulfurs. Now, in the case of angiotensinogen with this disulfide bond, they are liable or susceptible to being broken as the molecule changes from an oxidizing to a reducing environment. In a reducing environment, the bridge between the 2 sulfurs is broken and the 2 parts of the molecule can move apart. In an oxidizing environment, the 2 sulfurs are bonded and held together, and they hold the molecule in a more active shape, that is, will more readily release the hormone angiotensin that controls blood pressure.
If the angiotensinogen is in an oxidized environment, does that mean, therefore, it is more exposed and therefore more likely to be cleaved by the renin and for the hormone to be released?
That is right, that the molecule exists in 2 forms and they switch from one to another as the protein moves from a reducing environment to an oxidizing environment.
Why would, say, a therapeutic approach using this early, perhaps, mechanism be better than current, say, treatments for hypertension?
It needs to be emphasized that what we're looking at is the initiating stage in the release of the hormone angiotensin. There is a second stage in which the hormone is refined into the form that actually interacts with the arteries. And this second stage is controlled by an enzyme for which have now been designed a series of inhibitors that people may well know them as the ACE inhibitors, ACE inhibitors, that are used very effectively to treat blood pressure. But the problem is, one problem is, that the use of ACE inhibitors is contraindicated for the very best reasons in pregnancy. And it is in pregnancy that we perhaps get one of the greatest challenges of hypertension for people in the prime years of their life. The condition of preeclampsia, which is so common, it affects 2 to 7% of all pregnancies, is still not well understood. So what we've done is add a facet of understanding because we've shown specifically the change of angiotensinogen to the more active oxidized form takes place in preeclampsia. It opens prospects for the treatment of what has otherwise been a very resistant condition. That is the high blood pressure of pregnancy that is the accompanying and prime feature of preeclampsia.
So yes, I imagine this has a great deal of benefits if it were to come into practice. So I guess what actual stage is the research at now? How far away do you imagine we are from that perhaps being a therapeutic used?
The quick answer is that now we're beginning to understand the mechanisms. It opens various approaches, theoretical and otherwise, to treatment. And that could mean many years ahead. But there's been an intensive effort made to look at therapies for hypertension. And now as we look at the various treatments that have been tried or being given in trials, certainly in their early stages, there are some that we feel, in fact, might be relevant to the basic process that's occurring. And we hope our findings will be an encouragement to these people and eventually to pharmaceutical companies to follow this up.
Robin Carroll, Emeritus Professor of Haematology at the University of Cambridge. Now, that's it for this edition of the Diamond Podcast. But do join us again in the new year, 2011, for more insight into the science taking place at Diamond. In the meantime, though, if you have any questions about Diamond or the research taking place there, the email address is podcast@diamond.ac.uk. You can also listen to previous editions of this podcast online at diamond.ac.uk/podcast or nakedscientists.com/diamond. You can also subscribe to the podcast on iTunes. Thank you this month to Gwyndaf Evans, Yvonne Jones, Sarah Boundy, Jim Naismith and Robin Carroll. But until the new year, thank you for listening. I'm Mira Senthilingam.
The Diamond Podcast is brought to you by Diamond Light Source and produced by thenakedscientists.com. There's more information on our website at diamond.ac.uk/podcast.